thank you all for coming here today. So I would imagine that many people in this audience have been touched by Alzheimer's, one way or the other, friends, loved ones. This is a devastating disease for which there is no known cure. So Alzheimer's is a complex disease. It's thought to be caused by a combination of our own genetics and the environment that we're exposed to throughout our lifetime, including our lifestyle. And it happens when there's this perfect storm. It's a combination of formation of protein aggregates in the brain, known as amyloid plaques and tau tangles. And this is accompanied by activation of microglia, the immune cells in the brain. This leads to inflammation, neuronal injury, and then finally dementia, the symptom of Alzheimer's disease. So currently there are more than 7 million people in the United States who have Alzheimer's disease, and this number is expected to nearly double by the year 2050. So there's an urgency that's motivated my lab to take an approach that could potentially accelerate the search for Alzheimer's cures, and that's what we're going to talk about today. So my lab is in the Conrad Prebys Center for Chemical Genomics. This is a state-of-the-art drug discovery center here at Sanford, Burnham, Prebys. It's a really unique resource. So drug discovery is a multi-step process, and quite often it starts with screening through hundreds of thousands of chemicals using robotics. So once you find a chemical that does what you want, you go on to test it in animal models and then in clinical trials. So many important drugs have been discovered using this process, but it takes 10 to 15 years and can cost over $1 billion. Moreover, out of all drugs that go into clinical trials, only about 10% of them end up being approved. So there are a lot of improvements that are coming down the line to this process, including use of AI. But in the meantime, what my lab is doing is a more pragmatic approach known as drug repurposing. So there are thousands of drugs out there that have already been approved. We know that they're safe. Why don't we test these to see whether or not they could be repurposed as Alzheimer therapeutics? So to do this, we're using a technology known as human-induced pluripotent stem cells. So I could make a pluripotent stem cell from any of you, from a simple cell like a skin cell or a blood cell. So I take the cell, I turn it into a pluripotent stem cell. This means all-powerful cell. And I can use these stem cells to make virtually any cell type in the body. So importantly, I can also make these cells from patients. So these are neurons that have been made from Alzheimer patient induced pluripotent stem cells. And remarkably, they actually express a symptom of Alzheimer's. They have high levels of phosphorylated tau, or p-tau. Now this is the precursor to the tau tangles that we talked about in the beginning. So this was work from Larry Goldstein's lab at UCSD. So what we decided to do was we got together with the Goldstein lab and we asked the question, could we find an already approved drug that would lower this level of p-tau or phosphorylated tau in these Alzheimer patient-derived neurons? And so what we did was, again using robotics, we screened through more than 2,500 approved drugs until we found hits. And most of these hits were statins, the common cholesterol-lowering drug. And so this is what this looks like. If you take these Alzheimer patient iPSC-derived neurons and treat them with statins, you lower the level of phospho-tau, but the neurons are still just fine. So this study led us to another cholesterol-modulating drug known as efavirenz that's currently in a clinical trial in Amsterdam. So rather than 10 to 15 years and billions of dollars, this was about 5 years and billions of dollars. So I'd like to close here with another repurposing project in my lab that's funded by the Epstein Family Research Collaboration. So in particular, I'm involved in a program called the Powder for Pennies. This is the brainchild of Dan and Phyllis Epstein, and it's specifically focused on searching for drugs that can be repurposed as treatments for Alzheimer's disease. So at the center of this story is a region of your brain known as the locus coeruleus. It's right here at the base of your skull. So the neurons in the locus coeruleus, or the LC, send their projections out all over the brain and they release norepinephrine. Norepinephrine is really crucial for the health and function of many brain cell types, but these include neurons and microglia, the immune cells that we talked about before. So what happens in Alzheimer's disease is way before there are any symptoms detected, neurons in the locus coeruleus begin to degenerate. And we can detect this in brain imaging studies. So as these degenerate, release of norepinephrine is reduced, and this leads to inflammation and neurodegeneration that's thought to contribute to Alzheimer's disease. So is there a drug that could actually increase the level of norepinephrine to compensate for its loss in Alzheimer's disease? And there is. It's called atomoxetine. And the Powder for Pennies program currently has a clinical trial proposal to test atomoxetine on Alzheimer's disease patients. Now, in parallel, what my lab is doing is we're using Alzheimer's disease patient-induced pluripotent stem cells to develop disease-in-a-dish models. So we're recapitulating this whole system. From these patient stem cells, we've made the locus coeruleus neurons and microglia, and we're studying the way that atomoxetine affects these cells. So I want to end here on a positive note. There's really a lot of amazing research being done on Alzheimer's disease cures across the entire world, and I think there's reason to be cautiously optimistic. But I think we'd all agree that the cure really can't come fast enough.